So good morning, and welcome to this session on envisioning intelligences. This is 8 o'clock session Eastern Time in the US. I'm Steve Uzzo, and I'll be hosting this session. Over the next hour, we'll be exploring the unseen world from peptides to protists from an artistic viewpoint with Mara Hazeltine and how bacteria and yeast get along from scientist Rob Dunn. Stay tuned for that, for a lively exchange where we discuss perspectives and address your questions afterward. So we'll start the session with Mara, who's a renowned international artist in the field of sci art, as well as environmental activist and educator. Hazeltine collaborates with scientists and engineers to create work that addresses the link between our cultural and biological evolution. Her work takes place in the studio, lab, and the field, infusing scientific inquiry with poetry. She was a pioneer in the translation of scientific data and bioinformatics into three-dimensional sculpture, and became known for her outsized renditions of microscopic and submicroscopic life. Hazeltine's been a contributing member of the Explorers Club since 2008. She's been awarded Return of the Flag with honors for her work on the high seas with Tara expeditions, studying atmospheric climate change and its relationship to planktonic ecosystems. I'll share my screen now so that you can see her presentation.
Okay, this slide, just keep there.
Okay.
This is actually a diagram of from one of my favorite books, Bioethics: Bridge to the Future, which was published actually 54 years ago, but it has a diagram in it of how our biological uh, and our ecological or our environment, our cultural evolution are in fact completely linked. And this is kind of something that is a substrate to all of my work in that I think that art and the culture that we put into the world directly has an effect on our evolution as a species and our entire evolution, and in fact, as a biosphere. So, the following slides you'll see reflect this equation, and yeah. So, next. So, I'm on a mission to infuse art and poetry into the medical and field sciences. Next. So this, the original title of this is "Those Could Be Anything," and I think that that is pretty much the title of this talk for me. "Those Could Be Anything" refers to the fact that this is actually indeed a rendition of a blastocyst about 8 days after insemination, when the slides divide, but you can use them as stem cells. And then the title became Stem Cell Mandala. But those could be anything refers to the materiality of the science-based. You'll see I work a lot with proteins, which are proteins, but they are the substance often of viruses. So those could be anything. It's a lot about the organization of the material science, and but that's also the organization of how you perceive the work as art. Next, please. So this is outside of Cold Spring Harbor Laboratory, which is in Long Island, and it's the piece that I became very entranced with the notion of using bioinformatics as a scaffolding for sculpture and computerizing it. So this is a ribosome. Oh, by the way, this piece is about 20 years old, or maybe a little older at this point. So, this, and it still remains my largest piece at 84 feet. So, bioinformatics was pretty young at the time, but before, to create proteins, they used something called the Charles Supper wire bender, and it was just this machine where you would bend metal to the coordinates of a protein or a polypeptide. And this was a way that I developed to computerize that. And this piece is the part of the cell that creates the proteins called the ribosome and is clinging there to the endoplasmic reticulum. The project was conceived to be the entire cross section of a cell. So, proteins actually and viruses they have no color. So I added color. The protein actually next is in the far distance is that it's the beta lymphocyte stimulator protein, which was to fight AIDS and lupus, and it was sort of a shimmering color. Also, proteins are in a constant state of movement, and how to depict that as a stagnant sculpture. So one way that I used in Waltz of the Polypeptides to sort of create the fact that they're in these 3D environments is to do cross sections, like using a Japanese landscaping so that just little parts of them poke out and things like that. Um, so this was meant to be the golden champion and create these amazing vaccines. So I chose this gold color, and I use that a lot actually, and we'll see that more next. So this I actually happened to be next to it. This is a virus. This is the SARS virus. This is in Singapore. This piece was made in 2006, and it's called SARS inhibited, and the path in the stone in between the sculptures is the shape of the protease. And I actually have a model of it where I am right now. I could try and show it. Do you think that would be too distracting?
I know it's up to you. Um,
I think we can skip that.
okay.
But basically the idea is that the human could step onto the protease path and be the stop or the thing that makes the virus stop being able to interact with itself and thus stop SARS. So that I purposely made red and sort of angry looking, and once again I'm using the technique where you only see part of the virus because the entire virus it was just too complicated to make as a 3D sculpture. Next. So this is another big protein, and this is actually healthy, and this is for I call it homologous hope, and it's in Philadelphia, and there's a strand where you can see the there's LED lights that are actually this is a healthy protein, and it's repairing the damaged DNA in a healthy breast cell. And here again, I wanted it to look very hopeful. So, I mean, it's obviously a very complex piece, but I made it look kind of like a cloud or a unicorn even in pastel colors. And it's in this huge Raphael Vignoli atrium, and it's sort of meant to rise above and give hope. That was really important to me after visiting the hospital, which of course are places where people feel so destitute, and I wanted to create something light. And so here again, I did that through color and the shape. I mean, the shapes of the proteins, they look on what time I like scrambled eggs, but on the other time they are based, they're accurate. I'm a figurative sculptor, and I think that you can see or feel that in the work when you look at these ribbon diagrams. Next. Oh, so this piece I call Evolve, and it's really kind of similar to SARS Inhibited. I did it at the end of COVID, and it's never gotten the recognition that I feel like deserves. But the pearl represents, humanity's shared layers of knowledge that it took to create a vaccine. And here is, part of the virus. Um, and then I developed an entirely new technique to do video with this. Please roll them, Steve. Next. That shows it shows vibration. Our soundtrack could be better, so you're going to
Are
hear
you
this with
are
no
you hearing the
soundtrack.
sound?
Well, no, I took the sound off of it because
Oh, okay.
for various reasons I thought it could be, but we developed a way that if there is a sound, we can make it vibrate to the coordinates. So there's that. Next. Oh, I think we already saw that. Okay, so this is another kind of virus in a way. It's the virus of humanity. We are really are as an environmentalist that we're not showing. I'm kind of cut out most of my environmental work, but we're really our own worst enemy. And I did a lot of work on plankton, and I am, as Steve knows, a big fan of Ernst Haeckel and his beautiful radiolaria and single cell protists. And when I went with Tara Foundation in 2011 on their ship, I was looking forward to seeing all these beautiful plankton, but what I saw a lot was degraded plastic. And so I created a body of work depicting the plastic as kind of evil and creating disease, and the plankton as pristine and beautiful. And I made this reminded me of the opera La Boheme, so I made a film about that, and that's next.
Let me know if the audio comes through.
Okay.
Can you hear it?
No, I can't hear it.
La Boheme, a portrait of
Oh, I
today's
hear it.
in Paris.
Okay.
The tragic tale of interspecies love at the dawn of the Anthropocene age. The young bohemian poet Rodolfo from Puccini's opera La Boheme falls in love with Mimi, who is not a young grisette perishing from consumption, but instead a tintinid plankton ensnared in plastic and consumed with dinoflagellate parasites in an acidic winter ocean.
I don't hear the music though.
No music?
Oh yeah, just yeah. There I did hear it for a second. Look, let's just freeze frame it and I'll talk about it a little bit. I think it's too long. So to make the plankton, I also used uranium infused hand blown glass. But what I wanted to say really about that was the plastic was really roughly made, and um, the contrast really, I think, created the visual narrative of um, the plastic being quote unquote bad and the plankton being good, and it was glowing and beautiful, but they were mixed. And that's what you can really see in this video. But I think it's gonna eat into Rob's time too much, I think. So let's just move on past this video. So what do you see when you first see this, Steve? What would you say, or Rob, what would you say when what do you think this is?
So obviously the leopard it takes
Well,
front
you
and
know what
center.
that I didn't make that. I'm
But
talking about the piece on the right.
ob- obviously the thing hanging above it is very dramatic.
So yeah, no, so what I wanted to say is back to the those could be anything is, um it's actually a radiolarian skeleton, which is a single-celled protein, a protist. They're the most sort of plentiful in the ocean at about 96,000 times magnification. But when I first saw it after COVID, I put it up right before COVID. Um, I thought, oh my gosh, that looks like COVID, and so did many people. And that's because protests had been or COVID had been, in the news so much, and we had seen it so much that it really, influenced us. So I just thought that that was funny that, um, you know, under a certain largeness, smallness, there's virtually no gravity. So a sphere is a pretty normal way for um, something to arrange itself. So it's not, abnormal that there'd be a skeleton of a radiolarian, which is made of silica glass, and uh, a protein, which is a virus, arranged a similar way. But I just thought that that was kind of a unique perception shift. Okay, this is the last slide.
Yeah, and we should note that this is the gallery at the Explorers Club in New York City, in case you can't see the bottom. It's a pretty prestigious place to have something hanging, particularly at that scale.
It's yeah, and it really so it's in the trophy room, which obviously a lot of the conservationists like Rockefeller and stuff that were original explorers we're also hunters. And it was a different time when being a hunter, it wasn't like you were hunting the last spotted tiger. I mean, there was enough of them where they wanted to protect land because it was their hunting grounds, the same way Fidel Castro protects his fishing ground. But now, at a time when we're going through the mass extinctions, to have a skeleton there, I feel that it kind of unites the room, which is called the trophy room. It's at the top of the club, and yeah, you should go visit it, the club. There's a lot of interesting artifacts there in New
I'm
York
going to
City.
stop sharing now. We're done, right, Mara?
There's one more slide, I think.
Oh, okay.
This is a slide um, from my work, Ghost of the Zombie Sea, and the tagline in French is "Je mange la mer pour dîner." So it's about eating death for dinner, and kind of the concept behind it is that um, you know the biosphere will eventually survive us, but will we survive us? And corals may go extinct, but there are corals that don't need zooxanthellae, symbiotic algae, and they may replenish also. But will we? And that's what I wanted to, as an environmentalist, take a little bit of a left turn and my lecture with. But on to Rob.
Okay. Well, thank you so much, Mara. It's been great. And let me just get back to the Zoom here. So our next guest is Rob Dunn, a biologist, writer, and professor in the Department of Applied Ecology at North Carolina State University. Rob obtained his PhD in ecology and evolution from the University of Connecticut, where he studied the recovery of tropical forests in Costa Rica, Peru, and Bolivia, and clear cutting and the use of traditional agriculture after clear cutting. He sought to understand how long it takes for the biodiversity of animals to return to regenerating forests. He's written several books on his science essays, and his science essays have appeared in BBC, Wildlife Magazine, Scientific American, Smithsonian, National Geographic, and others. He's become known for efforts to involve the public and citizen science in arthropod surveys and bacterial floral studies. So let me, you're sharing your screen now.
I am, yeah. Can you see my slides?
I,
I'm starting with Mara's slide.
oh, okay.
Well, thank you so much, Steve, and thanks, Mara. I would, I'm going to jump into my talk, but I would sort of rather see more of Mara's amazing art. I wanted to segue from one of Mara's last slides, because what I want to come around to in the end of my talk is this recognition that while our conscious brains are just coming to terms with what it means to think about microbial worlds, that our bodies and cultures have had relationships to these smaller worlds for a long, long time. And so I think Mara's point that the perspective on this amazing sculpture shifted as our cultural conversation around views of the microscopic changed, that similar things have happened for a long, long time. And I was struck in a conversation earlier this week with Yvette Runninghorse, who pointed out that in Lakota, the word for microbes is little relatives. And I love this sentiment because it emphasized this relationality, that we are related to these smaller worlds. And so to think about these things, I'm going to zoom in on a strange, smaller world, which is the world of sourdough bread, which I think in many ways is a world that is like the world at large. And so with sourdough, I'll lead you to two bigger stories about the earth. One about the ways in which our unconscious bodies are negotiating vast terrains, and the other about the quasi-conscious intelligences of human cultures. This is a project that I'll talk about that has involved thousands of participants around the world, more than 40 collaborators, probably more like 100 collaborators at this point. And we all looked to get together at sourdough bread. And so it was this sort of object in front of all of us. And so it's really been a wonderful project. And the quarry is at once macroscopic, the bread, and microscopic. And so every sourdough bread that anyone's ever eaten has contained yeast, which make the bread rise, lactic acid bacteria, which make it sour, and acetic acid bacteria, which are not always there, but most of the time, and give it a different kind of sourness. And historically, this bread was a technology that allowed food storage, food transformation, food shipping, and water purification. And we asked people around the world to send us samples of their sourdoughs to get a sense of what's living in those bread making beings. And so we got samples from all of these places. This was a transdisciplinary project where the education was bidirectional, and invariably we've learned more from the participants than we have offered them back in some ways. And these starters were really different. And so some were stored in the fridge, some at room temperature, some were fed often, some rarely, some were more than 100 years old, some were in businesses, some were at homes. And we could then study all kinds of things about them through our collective, their composition. So what species is in each one of these starters used to make bread? What are the genes? What are the traits? What are the collective traits? And so we could see that while one sample might have the yeast Saccharomyces cerevisiae, if we looked across samples, many did, that standard bread and beer yeast, but many had other yeasts. And so there are these mystery yeasts people had relied on for thousands of years that we don't know very much about. We could do similar things with lactic acid bacteria and see that many of the bacteria, if you look at the up and down lines, each one is a sample. Many of the bacteria were Lactobacillus brevis, the sort of standard bearer of sourdough. Others were Lactobacillus sanfrancensis, the bacteria that in San Francisco people say makes their bread best and the holy bread of fancy food. But there were many, many, many other lactic acid bacteria. And so then we could figure out how do we explain this variation. We can make general predictions about it and develop general models to account for it, or we could try. And each person on the team wanted to answer a different thing and still does. And so Liz Landis wanted to know, are the functions of the starters predicted by the composition? So if you knew which bacteria and fungi were in an individual starter, could you say what its flavor was or how fast it rose? And so this involved doing all kinds of crazy things like having giant chemical sniffers for the breads and then having people trained to sniff alongside the sniffer. This on its own, this is an amazing talk, what's happening in that picture. And then we could see, if you look on the y-axis here, this is the percent of acetic acid bacteria, so vinegar bacteria in a starter. And the x-axis is the dominant note smelled from the starter. And so the more acetic acid bacteria, the more the dominant note, the dominant smell was acetic. And a lot of people don't like that. But we could predict that just by how much of this one kind of bacteria was present. Erin McKinney studied whether the grain type in the starter influenced the microbes present. And so she used an educator lens. And she did work with students to do experiments in classrooms to make hundreds of different starters with different grains. And what she found was that the composition of the starters depended on the grain, but so did the smells, and that the kids could smell it. The kids could sense these biological differences by using their sense of smell. We looked at whether the diversity of starters, how many species are in them, are predicted by elevation or climate. This is kind of the macroecologist question. It's what my ecologist people wanted to do with this. It's like what you would do with birds. And the short answer was no, that the starters don't obey the classic rules of biogeography, at least one set of rules. But is the composition of starters predicted by geography rather than climate? Yes, sort of, with some caveats. And so you can see from these different scientists involved in the project, different scholars, we're seeing these different things. Others wanted to know, where do the bacteria and yeast in starters come from? This was the participant lens. This is what the people at home wanted to know about their own starters. And in Korean, there's a concept, sonmat, which is hand flavor. And it's the idea that the person who makes food contributes with their hands to the food. Taken literally, we wondered, do the people who make sourdough actually contribute microbes to the sourdough? And does that influence its flavor? And so we did an experiment with a bunch of chefs, with a bunch of bakers from around the world in the Pareto Center for Bread Flavor in Belgium. We had them all make the same bread. The guy in the bottom right cheated. He pulled ingredients out of his pocket and produced that giant bread when he was supposed to make the same one as everybody else. But in the end, what we saw is that the baker's microbes went into their starters and influenced the flavor of the bread, which we could perceive when we sniffed the bread, right? You could sniff the smell of the baker in the bread. But the thing we hadn't expected is when we also sampled the bakers, and they were covered with starter. They basically had become their starters. And so the bakers had up to 70% yeast on their hands. And so by dint of their daily lives, they had become indistinguishable at some edge from the thing that they make. And this to us was a much more powerful result. Anne Madden, who thinks like a yeast, but also thinks like an evolutionary biologist, she wanted to know this other version of where did this bacteria and yeast come from? What's their natural habitat? Where were they 100,000 years ago? And that's a hat Anne made, inspired by one of the microbes that she works on. That's Anne under the hat. And so Anne studied this bigger origin. And what she found is that many of these yeasts we find in the starters actually originally come from wasps. And so their natural habitat, as others had intimated in particular contexts, is living in the guts of wasps. That's the only place they like to have sex. And then they get carried by the wasp from sugar source to sugar source. And so at some point, humans co-opted this wasp yeast relationship, and we became their wasps. We took over their role. And I'll come back to the bacteria. Matthew Booker is a historian. He wanted to know what makes some starters socially resilient. Why do families keep them for so long? How do they stay rescued? And so he studied his own family starter, which was passed on from Charles Bunnell. Charles Bunnell was the progenitor of Matthew's family starter. But when we did this study, we also had we asked people, is there anything else you want to tell us about your starter? And people wrote 70,000 words about their starters. And so Matthew went back and studied those words, and he found that part of what rescues starters, that keeps people saving them, is that people view them as members of their family. They're talked about using kin terms as living beings that are born, live, die, and are loved. And they're rescued because they're kept alive by multiple members of the family. And they're rescued by their status and social networks of which they are a part. And so there's this public historian perspective. Maggie Simon is a scholar of English literature. Amina Alatis Bradford is a theologian. They both became interested, and their perspectives were totally different. Maggie Simon wanted to think about what the 1600s and 1700s British literature said about how people were thinking about their starters and their family. Amina is really interested in thinking about, in the Christian tradition, when bread is the body of Christ, what does that mean about how Christians think about the microbes? What does it mean for those microbes? Are they then holy? What role do they play? And so what is the Christian tradition thinking about fermentation? And these projects have been amazing. I can't tell you the answer to either one of them, because the other part we've realized is that different perspectives move at different times. And so Amina needs to write a whole book, whereas the evolutionary biologist needs to write a slim paper. And so we have these incommensurateness that we make space for. The archaeologists want to think about, can we predict the starters of ancient breads and beers? And so we work with these two great archaeologists. The genomicists want to think about, where did these yeasts evolve and how did they move? And I can quickly say the whole literature on where the yeast evolved, Saccharomyces, is about Mesopotamia. In one of these meetings, we had a collaborator who happened to be Chinese in the meeting. And she pointed out that there's this whole starter tradition in China. Why aren't we talking about that? It had basically been missed from the Western English language literature. And it now looks like the evolution of the yeasts in our bread may actually come as much from China as it comes from Mesopotamia. And we'd been missing it entirely because of the narrowness of our lens. We now work on ancient beer, and these are wreaths used to seed a new beer in Norway, in this traditional Norwegian tradition of making beer. And Hannes Schrader and his collaborators and colleagues have been able to work with us, or we work with them, to identify and maybe actually find alive some of the ancient yeasts on these wreaths. A great part of this tradition is that when you make these traditional Norwegian beers, you have to yell at the yeast to enliven them. And so traditionally, you transfer these wreaths into the beer, and then you scream at them, "Please make me beer." Um, and so that one point here, which is a point I try to work towards in my entire life work, and I'm coming around toward a kind of end, um, is that diverse perspectives on this single universe of bread offer us something that a single perspective doesn't. And they start to show us a way, going back to that Mara's amazing piece in the Explorers Club, that we have to kind of look at what microbes are doing sideways and accumulate these sideways looks to see something that's more like a whole, or at least our conscious brains do. And I had this great quote, and I'm having a user fail here a little bit, that we're part of the Earth starter, as Ren puts it. We're not outside observers. We're all actors in a comprehensive drama in which humans and the non-human world take part equally. And I think that being part of the idea that the microbial is the microbes are our little relatives means we can't see well enough. And it's for this reason that to me, the art plays such an important role in anything we do in thinking about this world, because we're so much in it, but also not fully aware of it, that our ability to see is really limited. Now I'm going to briefly talk, watching the time, about ants and yogurt to pull this all back together so we've got time to talk together. A number of years ago, we became aware of Rasmus Monk's work at the Alchemist restaurant in Copenhagen. There's a longer story here, but the short version is Rasmus and his team were making little yogurt pastries, in essence, that were like ice cream yogurt, using ants to make the yogurt. I knew from a previous interaction with Sevgi Mutlu at the Rachel Carson Center that there are Turkic communities in Bulgaria where people use ants traditionally to make yogurt. We were able to pull the chef perspective and traditional Turkic perspectives in Bulgaria together to study with a big team led by Sevgi and Veronica Sinot, yogurt making in Turkey, or in Turkic Bulgaria and Turkey, and show that the ants are actually helping to create the yogurt. The microbes from the ants and the enzymes from the ants and the acids from the ants coagulate and flavor the yogurt. And the more we have studied this phenomenon, the more we've seen that embedded in the traditional knowledge of this yogurt making are all kinds of knowingness about how to do this to make it safe, that are not verbalized but are tacit in the process of putting the ants in the yogurt, putting the yogurt in the ant colony, and taking all of these steps. The other thing that we found is that in the mouths of these ants, and these little pockets they have that are kind of like the cheek pouches of some monkeys, is the only place we've ever found sourdough bacteria in the wild. And so we now think that somehow the sourdough bacteria originally get from the ants into the yogurt and make their way into sourdoughs. And so the strangeness of all of this is great. And so what I want to point out with the yogurt is that there's a lot of traditional knowledge all around us about microbial worlds that might not name them and yet is in relation to them. The other thing I want to say, that goes back to some of what Mara said at the beginning of her talk, is that if we go even farther back in time, our bodies evolved in relation to paleo fermentation, to paleo reliance on microbes. And I'll say it, but not show it in the interest of time, that we now know that about 8 million years ago, our bodies evolved the ability to break down alcohol 40 times faster than those of our ancestors. Our bodies evolved the ability to detect lactic acid bacteria and to stop burning fat when they detect them and to calm our immune systems. And our taste receptors evolved so as to like sweet and sour things. And so these are all ways that our subconscious bodies evolved to relate to these microbial worlds. And so here I'll come to my two strange conclusions. One, for hundreds of millions of years, the intelligences of our immune systems and olfactory systems, which have always been relating to things like sourdough bread, if not sourdough bread, the microbial worlds, have been negotiating intelligent decisions, pulling it to the theme of today. And that more recently, hundreds of thousands of years, our cultures have been accreting complementary intelligence related to these microbial worlds. And much of this has not been conscious in the classical sense, and yet nonetheless knowing. And then I'll stop here with a provocation, which is to say, I don't know how we would know if our immune systems were conscious. Can we rule out the idea that our immune systems and negotiating microbial worlds around us are fully conscious, but just think that our other consciousness, the one with which I'm telling this talk, is too stupid to explain things to? And so I'll stop there. Thank you.
Wow, thanks, Rob. Thanks, Mara. Well, my mind's been blown. This has been a great session, and I've learned a huge amount that I didn't know. I guess, my my interest has always been, okay, so nature created us. So how do we think about that, and what do we do to help other people think about how their role in nature really is really is important to understanding the world. And you've just answered that question to some extent. Thanks, Rob. So this has been an amazing trip through some microscopic worlds, quite literally, some of it under our noses. And now it's time for some discussion. And I have a couple of prompts, a couple of questions to ask you guys, but also for our audience, if you have questions, please put them in the Q&A, and we'll try to answer them during the time that's remaining. So I guess one of the things that struck me, both talking about biological entities, and it's always about biology going down from the chemical interactions straight up through how we negotiate reality. So obviously these ideas have been put in front of people. So in your case, Mara, these are these incredible constructions that, and then for Rob, this idea that we've really created this link to between food and people and bacteria and yeast. What kind of responses do you get from people? And I know, Rob, yours was this vast, sort of colloquium of people, but just in terms of this idea. And I don't know who wants to start, if you have any ideas.
I'll offer maybe a quick segue to the art, and then I would love to hear Mara's response to this, which is we did a lot of work on belly button microbes for years, and we imagined that would cause people to have better relationships to their microbes, that it was a playful project. Think about the belly button. And about 5 years into the project, I began to think we were having the opposite effect, that we were instead convincing people to go home and scrub in some Odyssean heroic way. And then we worked with the artist Joanna Rousseau, and Joanna Rousseau staged these interventions where she asked people to talk to their belly button microbes. And that provocation changed the conversation more than anything else we'd done.
Wow.
And so I think, I mean, I'll say, I think as scientists, we imagine we can change conversations, but it's what we're least trained for and least able to do. And so for me, the places we've best shifted what people think are with artists, with other people who have the ability to provoke, to intervene, and yeah, I walk away from today thinking differently about proteins because of Mara's work. And so Mara, do you have thoughts in that context?
Yeah, I think, well, as I said, the same sculpture or object could be perceived in a completely different way, even as a different species, depending on what's going on globally and culturally. And I think that like your work is very meaningful, and just learning how you have to shout at the yeast to get it to ferment. And I also think that there's so many rituals that we don't really understand. I was at the Museum of Modern Art yesterday even, and I saw a guard tell somebody to stand behind a radius away from the art, and what does that mean? What is the ritual of even entering a building made for art, and what's different from that than from public art, where someone who didn't necessarily go into an art environment and experience art, what does that mean for them? So I think that all this, the ritual around the yeast was really fascinating, and like, there's this huge machine, and then there's the sniffer. I love that. I am lost at your original question. Can you bring me back to
No, no,
that a little bit?
what kind of responses have you received about it? So, I'm thinking about, I have seen your
Oh,
sculpture in Cold Spring Harbor Lab.
What responses have I received?
Well, do people express surprise, curiosity? I mean, I'm thinking, well, one of the things that impressed me
Yeah,
most was I was able to see and I took pictures of it, of course, um, in Cold Spring Harbor Lab, um, the
yeah,
um, the enormous sculpture there, and the scale of it, and the ability to sort of traverse it as a viewer, as an
right.
experiencer, was really kind of remarkable to me. I'm just wondering, because of your work is so striking, whether are particular things people have really come out with and said, okay, this makes me think of something differently than I did before.
Yeah, I think, I mean, I am one of those artists who tries to seduce through beauty, or what I think is beautiful. And through that seduction, that dance, get the viewer to understand the science behind it, and then do something about it. So I think there's, like, the seduction, the viewer who knows nothing about the subject gets seduced into wanting to know about it, then, hopefully makes an action based on that seduction, whether to learn more about the science, whether to learn more. I do a lot of reef restoration for coral and oysters, to do more for coastal communities, in the small island states and other places. But it's a procedure, and it's sort of like courting the viewer into caring about nature, the planet, our body. So I see it, I guess, as a courtship with the viewer.
That's
And I
great.
do it on purpose. I make my stuff sexy.
Thank
What
you.
I think is.
So one other thing, I guess, is so thinking about what's next. Um, and I know that you probably haven't covered everything that you're doing now, maybe because of the topic of these talks. But thinking about the future, where would you want to go with these fantastic ideas?
Oh, is that for Rob or for me?
It's for both. Whoever wants to respond to that.
Well, I
If
want to
they
work
at
with
all.
Rob. That's one thing. I'd love to
Yeah,
do
the feeling is
something.
very mutual, Mara.
Yeah, so I would love to keep in touch with Rob and do something. I'm currently doing a lot with this new NGO that I started, that I didn't really talk about during this talk. A lot of I do a lot with reef restoration that does not use plastic or concrete, and is based on planktonic structures. So that's where I'm going to be putting my efforts into. Um, so yeah, so that's my next big trajectory, and I had a retrospective of that. I've been doing that for like 20 years alongside this other stuff at the New York Academy of Sciences last year, and I showed at UNOC which is the World Ocean Conference in Nice last summer. So what is Rob going to be doing?
Yeah, I mean, it's a great question. I have concrete projects, and I'll maybe answer the abstraction instead. I mean, so in a concrete way, we're thinking about those ants and their mouths and moving into those details. We have a project on the where we give little pills to. This is with Dinah Parker and Sandra Anderson in Copenhagen, where there is little robot pills you can give to an animal, and it takes a sample of their stomach juices, and so we can figure out what lives in the stomach. And so it's like going into the deep sea of the stomach. Um, but and in a more abstract way, I'm really interested in how do we think about longer time periods. Um, and so if I'm thinking about those sourdough starter microbes, to take the example of today, how do we tell the story of all the interactions they've had to come to this moment today? And so their ancestors have been in the mouths of ants, they've been other places, and we're seeing this one instantiation of their being. How do we better tell that more comprehensive story about what it means to be them?
Hmm.
Um, and I think ecologists and evolutionary biologists, my people, like we have a way to tell the atomized story of that. We can do an evolutionary tree of a microbe, and we can separately look at what the interactions were at different periods. But we don't have a good holistic way to talk about it. Um, and I'm interested in that. I don't quite know what to do with that. In that conversation I mentioned earlier, with Yvette Running Horse Collins, she also mentioned that in Lakota, and I'm not going to do it justice, but the horse is not the concept of the thing you're looking at now or a species of horse, but it's all horseness across time. And so if I'm thinking about the bread and beer yeast, Saccharomyces cerevisiae, what's it mean to think about all of bread yeast through time and that bigger story of it? And
Hmm.
I find that really interesting. And I like working in the blind spots of fields. And that to me seems like one of our blind spots.
That's fantastic. And
Yeah, I like the blind spot of fields. Yeah, that's good.
for the sake of full disclosure, I've been working on a team with Rob um, to to uh, to look at food webs, and I actually presented for the group on Wednesday to a very receptive crowd on who eats whom. So somehow it's relevant. But yeah, these are really incredible ideas, large, big ideas, and I think certainly, and I know Kadi's, one of the purpose of doing this thing is to stimulate collaboration. So I think there's some ripe areas for discovery here. We have a few more minutes, and we don't have open questions, but I have one more. I guess thinking about the you know we're in a crisis of science literacy right now, and it's beginning to affect human health and the environment, as we know. This kind of ignorance about what our relationship is to these things, and what would you want to tell attendees about, how do we break through some of this? And I know both of you are doing amazing work in breaking through this, but you may have other ideas that you haven't really explicated. If you have any additional ideas, it would be great to hear them.
Uh,
I'm glad to start in a pithy way, which is to say, first, I've been thinking about that question for a long time and have the sense that over the last couple of decades, we somehow failed. And so whatever we're doing now, it has to be different. That's my first. And so I'm trying and not always succeeding to bring to meetings, to conversations about this, a pause to say, let's not assume that what we were doing in the last few years is what we need to elaborate. And then the other thing that I'm not always great at, but I think that we collectively need to do, there's my dissonance, is to listen, and to listen iteratively and ferociously. Um, and we've seen this with the bread project, that people know things we don't know, but they don't always have a chance to tell us. And so how do we create those contexts in which we can listen and we can hear? And our systems aren't structured for that. And so um, that I think that's the biggest thing I keep trying to learn.
Mara, any comment? We have 2 minutes.
It's been a really exciting panel, and I do think that there's a real crisis in education for science, obviously, right now going on, and I think that it's important to keep going, no matter what, even in small ways, and and art may be a great way to do that. To keep teaching going and keep momentum despite and go team. And
Okay, well, thank you both. This has been an amazing journey. And thanks for whoever has tuned in to participate. We really appreciate you coming to this. And I believe, Kari, I believe this is being recorded. We would have gotten a prompt for that. So if this,
yeah,
sorry.
it says recording. I'd love a copy.
Yeah, well, after the event is over, I'm sure we'll be in touch and we can sort that out. But thank you, everybody, and I guess it's time to move on to
Yeah,
the
and
next.
Rob, I'm gonna email you like today or tomorrow. Okay.
That sounds great, Mara. Such an honor. Steve and Kati, thank you so much. Everybody else, thank you.
Okay,
Bye bye. Signing off.
enjoy the rest of your Sunday. Have a bagel.
Will in New York. I'm in New York, so I will. Bye.
Bye.